Hello everyone, and thanks for joining us for the in-demand tutorial today. The title of the presentation is Hearing in Color, Visualizing and Quantifying Molecular Markers with Photoacoustic Imaging. My name's Drew Heinmiller. I'm the product manager for our Photoacoustics line of products. The agenda for today is going to start with a summary of in vivo molecular imaging, a very brief summary, and then an introduction to micro-ultrasound and photoacoustics using the Vevo LAZR. I'll then move on to describing the guts of the presentation, which is going to revolve around an experiment that we did. So I'll describe the experimental protocol, and then we'll move into the tutorial portion where I'll describe how to use our software to acquire images and analyze images, first for in vitro photoacoustic contrast agent characterization, and then the in vivo photoacoustic contrast agent study. I'll end off with a tutorial on how to quantify the images and the signals that are derived in the first two portions. And then we'll move on to just a few examples of some applications of photoacoustic and micro-ultrasound imaging. So first of all, just a really brief study of in vivo molecular imaging. We start out with some kind of animal model, some kind of disease model where, in this case, you've got a crude representation of a sub-Q tumor, let's say, on a mouse. Then we've got a molecular target, so we want to find a molecular target within that disease model. Then typically we need a targeting agent, which will go to that molecular target and enable us to image it. We generate an image, something like this. This is, of course, an optical image using a fluorescent tag. And finally, there's the step of quantifying the signal that is generated based on that molecular targeting agent. Now, many of you are going to be familiar with some of these molecular imaging modalities, for example, PET and SPECT, MRI, and of course, optical. But there are some limitations and challenges with these modalities to be able to generate the data that you need for your studies. First of all, with PET and SPECT, it uses ionizing radiation. Some of the protocols can be quite complex and the reagents are expensive as well. Along with the expense point there, MRI can be quite expensive, and there can be issues with limited throughput with MRI. For optical, there are limitations on the depth and resolution that can be achieved, and this can also provide difficulties in quantifying the signal that's actually there. I want to talk about how we can use ultrasound and photoacoustics to get around some of these limitations of the other modalities, beginning with the animal model. Because ultrasound and photoacoustics is non-invasive, we can use the same animal as its own control for doing longitudinal studies. We can do imaging in real time. Using the setup we've got, we can do two things. We can get a fairly high throughput, as well as being able to allow interventional procedures such as surgeries or injections or other imaging during these interventional procedures, which is feasible with our system. And I'll show you a picture of what that can look like. Secondly, the molecular target and the targeting agent for photoacoustics. First of all, the agent we can use can be multimodality. So examples of some photoacoustic agents are things like liposomes down here, gold nanorods, for example, which can be tuned to absorb at different wavelengths, and we'll talk about that later in the webinar, silver nanoplates, and carbon nanotubes as well. A lot of these agents are designed to be biocompatible as well, so we can use them in these animal models with little to no toxicity. Finally, the imaging. With ultrasound, we can get high resolution and we can maintain that high resolution at depth. That way, we also get anatomical and functional data. And this ends up being truly a multimodality platform where we have the anatomical data from the ultrasound image, that's the grayscale image you can see in the background here, and then the functional data, which is derived from either Doppler flow imaging, microbubble contrast, or photoacoustic contrast imaging, which is what you're seeing in this image here. The quantification is is also helped out by having that resolution at depth as well. So we can quantify in 2D and 3D at depth and measure many different parameters. So we can measure flow, perfusion, oxygen saturation, and of course what I'll be focusing on here, the contrast agent signal from photoacoustic and other contrast agents. So I'll introduce our system, the micro-ultrasound and photoacoustic imaging system at this point. First of all, what is ultrasound imaging? Well, we use a transducer to generate a pressure wave. That wave travels through a coupling medium such as gel, goes into the tissue of the animal, is reflected back, and we listen to that sound to create an image such as the one you see here, where bright signals are strong reflections and darker signals are areas where the sound is transmitted more freely. Now, the advantages, I've gone over them here already, but we get our high resolution, which is maintained at depth. This is a real-time, non-invasive, and in vivo imaging modality. Now, the difference between our ultrasound system and a typical clinical system is really the frequency. A clinical system will run anywhere from 3 to 15 megahertz, where we're operating in the range of 30 to 80 megahertz. Well, what does that mean? If you look over here on the right side, you can see an image of a human fetus with a clinical scanner here, and then we look down here and we see an image that appears to be the same resolution as the image above it. However, we then realize that this is actually a mouse fetus, so we're looking at something approximately the size of a coffee bean or smaller. So we can get down to very, very high resolutions to be able to investigate our animal models and look at things like micro distribution and so on. So essentially the high frequency really means high resolution. We, of course, have many different applications. Some of our strongest applications are cardiovascular measurements. These kind of echo measurements are used in the clinic already, so they translate very nicely to animal models of disease. We can do left ventricle analysis for doing things like cardiac output, ejection fraction, fractional shortening, and so on. We can look at blood flow velocities over time to look at E to A ratios and so on. And we also have software that looks at strain and strain rate. So strain in the myocardium, but also strain in the vasculature to be able to quantify vascular stiffness. There's a lot of we have to offer as well on the cancer side, where we can do things like early detection and screening because we have that high resolution and we can see very, very small lesions. We can look at vascularity by virtue of our Doppler imaging capabilities, and then look down to the capillary level at perfusion down here, where we can use ultrasound contrast agent, these small microbubbles, and do a tail vein injection and see the perfusion in a tumor or a given organ. And then, of course, volumetrics is a big one, being able to measure, because we can see with high resolution at depth, we can precisely measure things like tumor volumes. Now I'm going to talk about photoacoustics. So if you're not familiar already with it, it's essentially a hybrid imaging modality, a hybrid between optical and ultrasound. So we used a pulsed laser light to illuminate a tissue or an object. Anything that absorbs that light will heat up a little bit and give this thermoelastic expansion, which results in a pressure wave which transmits through the tissue and comes back to our ultrasound transducer, and then we detect that and we can create an image from that. Now that's the long version of the explanation. The short version is light in, absorption of that light, and then sound out. This is an example of the kind of image that we can generate using photoacoustic imaging. So what we're looking at here is a generated 3D image of superficial vasculature in the abdomen, and then an image of vasculature within the ear of the animal. And you'll notice that, I say vasculature because this looks like vasculature, and indeed it is. The reason that we see vasculature with photoacoustic imaging is hemoglobin absorbs light at the wavelength range that we're using. So we're able to visualize hemoglobin and therefore see the vasculature within the animal. Now, this has many different implications for being able to see vascular structure, but because oxygenated hemoglobin and deoxygenated hemoglobin absorbs differently at different wavelengths, we can use multispectral imaging, multi-wavelength imaging, and actually calculate how much oxygen the hemoglobin is carrying. And this has implications for a lot of different applications. For example, for tumor hypoxia. This is an example of a sub-Q tumor with a highly perfused region showing a higher oxygen saturation, and then a region that doesn't show much blood, but the blood that is there is showing a much, much lower oxygen saturation. So this goes to imaging of tumor hypoxia. Related to that is ischemia and angiogenesis imaging. A commonly used model for this is a femoral artery ligation. So the femoral artery of the animal is ligated, and then the regrowth or angiogenesis that goes on there, the regrowth of the vessels can be imaged over time. And then we can look at, how much oxygen is actually being delivered by the blood to that limb versus the control limb, which is this one here. There's the control, and there's the treated limb. These are 3D images, by the way. This also has implications, of course, for neuroimaging, things like stroke, where if we want to see ischemia in the brain, we can do that as well and measure oxygen saturation and measure the extent of ischemia. Of course, today I'm going to be focusing mostly on the molecular imaging aspects, the photoacoustic contrast imaging aspects, a few applications of which I'm going to talk about at the end of the presentation. So this is what the system looks like. Over here on the right-hand side, you've got the Vevo 2100. That's the guts of the ultrasound system. Beside it, you've got the laser. So all the different components of the laser, including the cooling system, the power supply, the optics, energy monitoring, everything is built into this box here. And we deliver the light through a fiber optic cable and right through the transducer itself. And then we can collect the ultrasound via this cable here. This is an enclosure that we can use to do the imaging so that this imaging modality can be used in the same room as other imaging modalities without any risk to the user's eyes. You just close these doors. You can, of course, still monitor your animal because we use this imaging platform which monitors the breathing and respiration of the animal and records it along with the images so that we can monitor how the animal's doing, monitor the physiological condition of the animal. It's also heated just to keep the animal warm during the imaging session. Now, the most important aspect of this system is that we use a tunable laser, so we can image from 680 to 970 nanometers. And that allows us to do multispectral imaging and thus do some unmixing as well. And that's going to be the focus mostly of this presentation. The software that's associated with this, we have been continually developing. There's going to be a new beta release coming out this fall. And those are the features that I'm going to be using for this tutorial to show you how the new software works. The real focus of this software development has been on a streamlined workflow, and we're coming at it from an applications perspective. So instead of having kind of a completely open system where everything has to be done manual, we're trying to really streamline the workflow with an eye to how to characterize the contrast agent that you happen to be using, and then how to collect images and quantify the signal that's derived in vivo from that contrast agent to do your molecular imaging. We've to enable this, we've integrated infusion pump control, so we can actually do the infusion of the contrast agent while we're doing the imaging and control when that's happening, and I'll describe that. And then, of course, really focusing on the spectral unmixing. The acquisition, the multiplexing, and the quantification of spectral unmixing has been vastly improved from the existing software version. And then there are other things like improvements to the graphing capabilities, which allow you to really look at your data in a much more flexible way and to be able to customize it and export that data easily. Okay, let's start talking about the actual experiment that we ran for this tutorial. The contrast agent that we're using, I'm going to start there because there are many different kinds of contrast agents, and one class of contrast agents that can be used with photoacoustics as well as some other imaging modalities are nanoparticles. We work together with a company called Optimios, who has developed or licensed the technology that was developed at Princeton, which is based on a polymer photoacoustic dye. So they create these dye-based nanoparticles. They're organic, they're biocompatible, and they can be functionalized with things like peptides and proteins and so on. The nice thing about them as well is that they can be customized so that we can have differing absorption spectra. So essentially, this is what it's going to look like, the nanoparticles that we're using from Optimios. One is called PAR 788, one is called PAR 900. The PAR 788 has a peak absorption wavelength at the shorter end of the wavelength range. The PAR 900 has a peak absorption at the longer end of the wavelength range. Essentially what that means is they're different colors. They look different, so we should be able to distinguish them apart from each other even when they're administered to the same animal. The second thing is the PAR 900s are folate targeted. So they're targeted to the folate receptor, and we're hoping to see that this targeting actually enhances the retention of that nanoparticle within a tumor which overexpresses the folate receptor. And that's a KB tumor type. So just to reiterate, the goals of this study really were to image, and the goal of the tutorial really is to show how to image multiple photoacoustic contrast agents in vivo in the same animal. And a sort of side goal of that is also to assess the folate receptor targeting ability of this nanoparticle from Optimios. I'm going to describe the protocol for doing the experiment now. The first step is to actually characterize the contrast agent in vitro. So we put the contrast agents in a phantom and we save the curves in the software. Now remember, I'm just giving a summary here, and I'll describe these steps in a little bit more detail later on. The second step is, of course, the in vivo experiment, where we're going to do a scan before the injection. We'll inject the PAR 900, that's the folate targeted particles. We'll do another scan just after the injection of those particles. We'll inject the PAR 788, the negative control essentially, the non-targeted particles, do another scan. And then we'll look at the 24-hour time point, the 72-hour time point. And once we've acquired all this data, it's time to quantify it. So we're going to spectrally unmix all the different signals. Now there's many signals in there, namely the blood, the oxygenated and deoxygenated hemoglobin, the melanin, and then of course our two nanoparticles. We're going to draw 3D ROIs and quantify all of that. Okay, let's jump right in. So let's start with the photoacoustic contrast agent characterization. So what we've done here is we've built a phantom. Now this is a phantom that we use for assessing different kinds of contrast agents. It's essentially just a frame on which we've mounted polyethylene tubing, and we just inject the compound into the tubing and then image this tubing in cross-section. I should say that we are developing a phantom that we're planning on making available to our customers. We're still working on that, and please keep your ears open and check our website in the fall to see how we're making progress on that. But back to the imaging. So we fill this container with water. We immerse the transducer, the end of the transducer, into the water and image the tubes in cross section, and that's what you're seeing here. So this is the cross section, just the ultrasound cross section of these tubes. So we've got 5 tubes in here, and we can see, you're seeing the double line there. We've got the sort of outer diameter here between the larger outside lines, and then the inner diameter of the tubing between the inside lines. Here's a scale just for a bit of reference here, from 8 down to 12 millimeters. There's also a guide we've applied to the software where you can properly line up. The depth of imaging is quite important, so you can properly line up your phantom so that you can do this reproducibly. Now we'll go into the acquisition. So in this case, we're going to use what's called the spectro acquisition. Basically what that does is it just sweeps across the entire wavelength range of the laser from 680 to 970, and you can choose the step size here. So in this case, we've just chosen a step size of 5 nanometers, but you can go right down to 1 nanometer. And this is the image you end up getting, where we see signal coming from each of the different samples that we've injected into that phantom, into those tubes. The next step is to characterize or quantify what we're seeing within the phantom. So we've got our image over here. We draw these ROIs around the different areas of the tubing, and that generates these curves here. So on the x-axis, you can see we've got wavelength down here. On the y-axis, you can see we've got signal intensity. Now I've chosen just to display these two here. So we're using these two nanoparticles. So these are the nanoparticles that I talked about earlier, the PAR 788 and then the PAR 900s. The green is the PAR 788. Now you'll see here, as I described earlier, you have a peak absorption, which is in the shorter end of the wavelength range, and then that drops off as you get to longer wavelengths. The PAR 900, the folate targeted particle, does absorb across the entire wavelength range, but its peak absorption is out here in the sort of 800, 830 is where this sort of plateau begins, 830 nanometers. So now we know the spectral characteristics of these contrast agents. The next step is to be able to save this into the software so we can use it to perform the unmixing later on. So that's what we've got up here. You've got if you click on the Save Unmix button, it takes you to this panel here. So now we've got the region that we've selected. This is the region that has the PAR 900. We've got the group, and I've just customized it. So I've called it the Optimios group. Of course, you can have a subgroup. I've named the component and chosen a default color for it as well. So now once I hit save, this is going to be saved into the software. And if you then click on Preferences and look at the Spectral Unmixing tab over here, you'll notice that the curves that I've saved under the Optimios group are the PAR 788 as well as the PAR 900 folate targeted. So now the spectral characteristics of those components that I've generated based on my in vitro phantom imaging are saved into the software and can be used for subsequent unmixing. Let's get to the in vivo portion now. We've characterized our contrast agents. Let's see how they look in vivo. When we start the in vivo acquisition, we have to consider a few things. First, we know we're injecting our different nanoparticles, so we know we're injecting our PAR 788 and our PAR 900, and we have to consider which wavelengths we're going to acquire in order to be able to unmix those. But of course, since we're in the in vivo situation, we also have other components to contend with. So there's the melanin, deoxyhemoglobin, which is this curve here, and then oxygenated hemoglobin, which is this curve here. So all of these components with their given spectral profiles will be present in the in vivo situation, and we need to be able to acquire enough data to be able to unmix those all and then subsequently quantify them. There's a couple ways to do that. The first way is kind of, it exists already. We've enhanced it a little bit, but it's the nanostepper mode here. So this allows us to collect, at this point, as many wavelengths as you want. And you simply type them into this box here, and it will just acquire one frame per wavelength and just keep cycling through them. In this case, I've selected 7 different wavelengths. And you can see that those wavelengths, 680, 710, 730, and so on, they correspond to the peaks and troughs of the different components. So I'm trying to choose things on the peak and off the peak for each of these different components. Now this is, again, a manual process. I've selected enough wavelengths that the unmixing should work fairly well. But this isn't necessarily the best way of doing it. Just picking your own wavelengths. There are specific wavelengths that will work better to distinguish the different components. We've tried to take the guesswork out of that by adding this new acquisition method, which is the unmixing acquisition method. And this is the one that I would recommend with this new software. to do any kind of spectral unmixing where you know the components at least that you're using. So in this panel, you'll notice that instead of selecting wavelengths, we actually select the components that we want to unmix for. So in this case, I've selected, these are the VSI system components. They're built into the system already. Blood, for example, so deoxy and oxyhemo, I've selected those. And methylene blue is another one. Now, why haven't I selected my components? The reason is this feature was not available when I did the experiment. So I was choosing my own wavelengths with NanoStepper, but if I were to do this again, I would use the unmixing panel and I would select my PAR 788 and my PAR 900 components. The software would then determine which wavelengths it should collect in order to perform the unmixing. So it automatically selects your wavelengths for you, takes the guesswork out of it. It selects the wavelengths that will work best to be able to distinguish these components for each other when you're trying to unmix them. You can, of course, save presets to make this simpler and not have to manually check these every time. But once you've done this unmixing, you can start the acquisition. You can choose more wavelengths than the minimum. It just automatically chooses the minimum to have the fastest scan. But you can choose more wavelengths, and it will simply add wavelengths onto the ones that have already been chosen. Okay, let's start the experiment here. So we imaged the animal already before the injection because we want to have a baseline. Now we inject the PAR 900, the folate targeted particles. Now to perform the injection, in the past, you've had to open the enclosure. You can't actually perform the injection with the enclosure open. The laser won't fire. So you'd have to image, open the enclosure, do the injection, close the enclosure, and then continue with your imaging. But we've got this infusion pump which allows us to do remote injections. So we can put this portion of the infusion pump, the injector itself, we can mount it onto the injector within the enclosure. Okay, so the injector mount is for image-guided injections. We can even perform image-guided injections using photoacoustics. In this case, I haven't. It was just a tail vein injection of this contrast agent. So I've got our tail vein catheter attached here, and then the infusion pump right here mounted onto that injection arm. So I can actually perform the injection while I'm imaging. Another nice thing about this is the integration of the infusion pump controls into our software. So instead of having to use the external unit, we can use our software. And there are lots of different parameters to use. What I'm showing here is just one of the defaults which is loaded into the system, which gives you your syringe diameter, volume, and so on. And we can perform that injection now using these different parameters. Of course, you can customize it and use your own parameters as well. Another thing that I want to point out here is what we'd like to call these event markers. So this was a request that we had many times from different customers where they wanted to make note on the cine loop or during the acquisition which particular frame an event occurred. So we decided a good way to implement this is to actually use the event marker as the initiation of the injection. So we're imaging, we acquire a few frames, and then we click a button on the system, and that sets a marker on that particular frame and begins the injection as well. So that's what these things are down here. When you look at the buffer here, you see these little dashes that go through the buffer. Those are the event markers. In this case, we had one marker to start the infusion, another marker to stop the infusion. Information about the volume that was injected and the time it took is saved with that marker, and then of course you can add other annotations as well to be able to know what kind of manipulation you did at that time, what injection you did, if you changed the breathed oxygen concentration, for example, or any other event you can mark down there. So that's a really nice feature that I'm happy to see integrated into the new version, new beta version of the software. Okay, now with our bolus injection, so we use the infusion pump. We have the marker there. remember we were acquiring with the nanostepper mode, so we're acquiring these different wavelengths, and this is what the cine loop ends up looking like. So it's something like this, and you're seeing a flashing in the image, and that indicates the switching of the wavelengths here. So the wavelengths are switching through those 7 wavelengths that I showed earlier, and this is the kind of image you get. Now this image is actually during the injection. So the injection is occurring right here. There's just before the injection. Now the injection is occurring here. And you may see some subtle changes in the image itself upon injection of that contrast agent as it goes into the circulation. But the better thing to do is, of course, to be able to quantify or to see, you know, in the signal intensity numbers, is it going up and so on. So we can draw regions of interest right on here, and then we can graph them. So in this case, instead of having the wavelength on the x-axis, you've got time on this axis here, and then you've got the signal. Now this looks kind of jagged. The reason for that is, remember, this is a nanostepper acquisition, so we're acquiring 7 different wavelengths, and it's over time cycling through those 7 different wavelengths, and then you can actually see the injection happening here. One thing that's been added to the software here is the ability to now separate the components. So I don't want to see the cycling of the wavelengths. I want to see each individual wavelength. Remember, we've got these wavelengths here that we've been acquiring, and we click on the separate components, and we generate these graphs where each component can be selected here and displayed. Again, over time, we're seeing each wavelength over time, the signal intensity at each wavelength over time. Now, I've only chosen to display 2 of the wavelengths, 680, which is here in this, the line with the circles, and 830, which is this line here with the triangles. Now, you'll notice that the enhancement of the signal at 680 here is much lower than the enhancement of the signal at 830 here in both of these ROIs. The reason for this is, remember, our curves. So we're injecting the PAR 900, that's this component here. If we look at 680, the signal intensity is much lower than it is at 830, which is the reason we see a much greater enhancement at the 830 wavelength. Now that's all fine and good, but we really want to quantify the component itself, not just the signal at the peak wavelength. How do we do that? Using spectral unmixing. So if we click down here in the bottom left panel, you'll see this dark side of the moon kind of icon here. If you click on that tab, it brings up this panel here. This is the spectral unmixing panel. Now what you've got listed here are the different components. So remember, I saved these components into the software from my Phantom, and I've got them right here, the 788 and the PAR 900 folate targeted. I've got a curve that I saved for melanin, and then a system curves for deoxy and oxyhemoglobin. So when I click down here, I click process. I then get an image like this. Now this is now been loaded into the multiplexer. So if you look over at this panel here, this is the multiplexer panel. This is where, again, we've tried to improve the workflow. This was possible on some versions of the previous version of the software. However, we've really tried to streamline the workflow here. We've added the components automatically. They each have their own layer, their own color. And we can adjust a lot of different settings, which I'm not going to go into here, but this is, again, something that we're trying to automate so that as soon as you unmix your signals, you end up with an image with your unmixed data. So now, remember, instead of showing wavelength, in this case, we're showing the different components because we've unmixed them. So we've got deoxyhemoglobin in blue. You're seeing it in a lot of different portions of this sub-Q tumor here. We've got oxyhemoglobin in red, melanin in this sort of pinkish color, PAR 900 in gold, and PAR 788 in green. Now, this image was taken before the injection, so you're not actually seeing any PAR 788 or PAR 900 signal. This is pre-injection, so you're just seeing the signal from melanin, oxyhemoglobin, and deoxyhemoglobin, even though we are unmixing for these components. So this is what we would expect. we're unmixing for the components, but they're not there yet, so we don't see them yet in the image. This is the bolus injection again. This time, instead of separating the components by wavelength, we've separated them by what the components actually are. So in this case, I'm only displaying the PAR 900 folate, and we're seeing the bolus injection of that PAR 900 folate in this ROI here. And you can actually see in the image, which has been unmixed, you can see that the PAR 900 signal, that yellow signal, comes into the image, and we see this enhancement of the signal. We can see how quickly it's being infused, and to the extent at which it gives us signal here. So how much of it is being infused? How much signal are we getting from that component? I've put these images here just as a really quick summary. So before injection, after the PAR 900 folate injection, and then after the PAR 788 injection. So you've got all three components finally in this last image here. Now we want to be able to quantify this in 3D. So we can quantify it during the bolus in 2D, that's fine, but we want to quantify this in 3D. So I did 3D acquisitions of all these different pre-scan and post-scan, and this is what it ends up looking like. I'm able to draw a spline, and I always use the BMO, the ultrasound image, to draw these because the anatomy is so clear on these images and high resolution that I'm able to draw these 3D ROIs on there. And I draw a few contours. It will interpolate between those contours to draw that sort of morphing ROI that you see there in the image. So we're able to see the borders of the tumor and compare it to where the skin is, and we're able to draw that 3D ROI. Finally, what does the quantification look like? After we've drawn that 3D ROI, the numbers are all displayed for the components that we've chosen to display. So I've only selected the PAR 788 and the PAR 900 components, mostly just to keep it a little cleaner on the screen. But you can see here that in this particular slice, you've got these two components, PAR 788 and PAR 900, but you also have the 3D quantification of those two components. Now you can see here that in that one slice that we're currently looking at, you see that the PAR 788 is giving us quite a strong signal, higher even than the PAR 900. But if we look in the 3D, we see that the PAR 900 is slightly higher than the PAR 788 signal. Now I've done the quantification here. I'm not going to show you that the individual ROIs that I've drawn, but we can draw those ROIs immediately after the injection, but then at our different time points. So in this case, the 24-hour time point and the 72-hour time point. And we can see here in the image that the signal from the PAR 788, the signal in green, is decreasing over time, whereas it appears that the signal in yellow, or gold, that signal is increasing over time. And I've actually just included the quantification in a chart here where we have our time point just after injection, where you've got the PAR 900 and the PAR 788 in 3D quantification, remember, and then 24 hours later and 72 hours later. So ta-da, this is the summary of all this data that we've collected, where we see that the PAR 788 does give us a signal. It increases after 24 hours and then goes down even below that sort of baseline after 72 hours. The PAR 900, on the other hand, does increase, similar to the PAR 788. It decreases after 72 hours, but it never comes down below that baseline signal, indicating that it is staying within the tumor to a greater extent than the PAR 788. And you can even see that in the image here, where you've got the PAR 788 signal really seems to be restricted to the skin line there, whereas the PAR 900 signal is throughout the tumor. So this indicates that it's possible that targeting that we had, the folate targeting, is actually helping to retain that particle within the tumor, and we can image that, essentially imaging that folate receptor. So this is only one animal, I should say. So I don't want to make too many hard conclusions about this, but this is a nice experiment where we can use the same animal over time, and we can also quantify two different components within the same animal: one control, the negative control, the PAR 788, and then the folate targeted, the PAR 900, all within the same animal. I want to conclude by showing you some applications for photoacoustic imaging in molecular imaging, but also branching out into other types of molecular imaging as well. Now, I know we've been talking about nanoparticles as contrast agents for this tutorial, and in the case of the Optimios particles, those dye-based particles, but I want to mention gold nanoparticles for photoacoustic imaging here as well. Gold is a great material for doing photoacoustic imaging. It absorbs light in the near-infrared range very, very well. It's biologically inert, and by changing the size of the gold particle, you can actually change the peak absorption wavelength. So similarly to how the dye works, you can change what those particles look like, and therefore use sort of multiple particles in the same animal. Our partners in this are nanohybrids. So this is a company that spun out of a lab at UT Austin that makes their own, in this case, silica-coated gold nanorods. They have the silica coating to make them more stable and to increase the signal strength, and they also functionalize them with antibodies, and they're working on other functionalization as well. So these can also be used for photoacoustic imaging. If you want more information about these particles, go to nanohybrids.net. They've got lots of different information about them there. And the reason I wanted to mention them is because there was a paper published recently in Cancer Research that takes advantage of this kind of technology, this gold nanoparticle technology. And this is actually the same lab where nanohybrids spun out of. It's Stanislav Milyanov's lab at UT Austin. And this is a paper that they published recently using a gold nanosphere in this case, and they've functionalized it with an antibody here. Now, this antibody is EGFR targeted. And the first thing they did is, somewhat similar to what we did in our experiment here today, which is look in vitro first. How do these things behave in vitro? So they had three situations where they imaged. They imaged essentially just cells in a dish with no nanoparticles. Next, they imaged cells in a dish with particles that were not targeted, so they're just floating around outside the cells. And finally, they imaged cells in a dish with their targeted particles. The targeted particles, of course, are taken up and endocytosed by the cell, whereas the non-targeted ones just, again, as I say, float around outside the cells. Now, they did similar to what we do, which is run this sort of spectro function or image across the entire or a whole wavelength range. And they note that in the situation where you have no particles, they get this relatively flat absorption spectrum. When you have the particles just floating around, you have a peak out here at around 550. And when the particles are targeted, and this is the key, they absorb, they actually redshift, so they absorb at a longer wavelength range once they're endocytosed by the cell. Thus, they can be identified, and they can be identified in the following way. They developed a tumor model, a tongue tumor, which metastasizes through the lymphatics down to the cervical lymph node here. This is a ventral surface of a mouse, by the way. You can see the mouth up there. So the metastatic cells go to this cervical lymph node here, and they wanted to be able to see, can we see micrometastases in this lymph node using this gold nanoparticle system? So they had a metastasis, which they were able to identify with fluorescence. And then with photoacoustic imaging, they did the imaging, and they discovered that if you've got your EGFR targeted particle, they inject the EGFR targeted particle at the tumor site, it drains through the lymphatics, and it will be endocytosed by those cells, and you can unmix based on that red shifted signal, that longer wavelength signal that those particles will generate because they're endocytosed by the cell. If you have a mismatch, so if you have your particle which is not functionalized, you don't see that redshift, and when you unmix for that, you don't see any particles. And of course, if there's no tumor, despite having your targeted particle in there, if there's no tumor in there, they're not endocytosed, and you won't see that red shifted signal. And that's what we're seeing here in the last slide. And of course, this can be done in 3D as well. So I see this as a very nice example of using nanotechnology, in this case gold nanoparticles, to be able to do functional imaging and really find these micrometastases which you would not be able to see otherwise. The next application I want to talk about is a little different. It's stem cell tracking. Now, this is work that was done at Emory University. These images are courtesy of Dr. Young-Sup Yoon's lab there. And they have a hind limb ischemia model, which I talked about earlier. They have a hind limb ischemia model where they ligate the femoral artery, and then they want to see angiogenesis, and they want to use stem cells to be able to enhance that angiogenesis and help recovery of that limb. In this case, they had two different cases. One where they had cells which were enclosed in a scaffold. So they enclosed these stem cells in a scaffold and implanted them into the hind limb of the animal, the ligated hind limb. And then they had another animal where they just injected these cells. They're just kind of freely floating, not enclosed within a scaffold. They injected those in another animal's ligated femoral artery hind limb. Now, before injecting these cells, they labeled them with something called dye-R. It's made by Life Technologies, which in turn is owned by Thermo Fisher. This is a lipophilic carbocyanine dye which integrates itself into the plasma membranes of these cells and stays in there. So these labeled cells are applied to the hind limb. And even though this dye-R is a fluorescent tag, it's also visible with photoacoustics, as we saw here. So we image them in a phantom, we see the peak wavelength, and then we unmix based on that spectral signal that we saw, similar to how we did this experiment. And we saw that the scaffold enclosed cells really do remain within this scaffold in this case, whereas the freely injected ones sort of spread out to a much wider area. You can see in the 2D as well as in these 3D renderings here, which have been unmixed for the dye-R as well as oxygenated and deoxygenated hemoglobin here. Now we can add information using the same system. We can add more information and gather more data about this particular application. So we can look at the same animal, the cell-injected limb versus the non-injected or contralateral limb. Now we can see not just the tracking of stem cells with photoacoustics, but we can also see the oxygen saturation of that limb with photoacoustics. We can see blood flow with the Doppler imaging and perfusion with contrast imaging. So now we've combined all these different modalities which are available on our system together to learn much more about this animal model than just where the cells are. We can learn about how they're affecting the oxygen saturation, the perfusion, the flow of the blood, the angiogenesis that's occurring in that ligated limb versus the non-ligated limb. So this turns out to be really, truly a multimodality imaging platform here. Speaking of multimodality, I want to talk about another kind of molecular imaging, this time reporter imaging. We want to be able to see a molecule of interest, and reporters can be good because we don't need to inject and rely on the delivery of a contrast agent to a given target. We can express it, in this case, inducibly. This is work that was done with Roger Zemp at the University of Alberta, where they developed a model where they had inducible tyrosinase expression. Tyrosinase is the enzyme responsible for melanin production, and melanin shows up with photoacoustics because it absorbs at these shorter wavelengths, and it shows this kind of linear decay as you go down to these longer wavelengths. So because melanin shows up, if we inducibly express it and we put the promoter for our gene of interest in front of it, it will be expressed along with the gene of interest, and we'll be able to do this reporter gene imaging. So they sent us these animals and we imaged them with photoacoustics. Before induction of the tyrosinase expression, over here, and then over to the left there, and then after induction of the tyrosinase expression over on the right. And you can see clearly on the bottom panel there that the tyrosinase positive tumor after the induction shows a huge amount of photoacoustic signal, which of course when you cut the tumor open you see there's plenty of tyrosinase expression there. And this is in turn confirmed by histology as well. Now this was published in a paper in Scientific Reports. Roger did his own spectral unmixing and showed the quantification. You could actually quantify the amount of melanin that was expressed. So again, this just shows this reporter gene, inducible reporter gene imaging, which could be of interest to people who are not currently using injectable contrast agents. So this is a paper that was also published in Scientific Reports, actually just a few months before Dr. Zemp's paper. It also used this was a group at Stanford, by the way. It also used tyrosinase as a reporter gene imaging system, but it emphasized the multimodality aspect of this. So photoacoustic, MRI, as well as PET, to be able to do this sort of triple imaging modality. Now, the thing I want to point out about this, I don't want to go into too much detail, but I do want to point out the different images. In the case of the MRI, you see the entire cross-section of the animal here with the tumor appearing up here. For the PET, you see a big longitudinal section of the entire animal here with the tumor appearing over here. But with photoacoustics, using our system, you can see that we're looking at just the tumor here, and we can now actually see micro distribution. We can see where in that tumor this gene of interest is being expressed. As all cancer biologists know, there's a complex environment. The microenvironment of the tumor is extremely complex, and we can now see the expression of different genes in, say, hypoxic versus non-hypoxic regions or other different microenvironments. So this, really lends itself well, this system really lends itself well to this multi-modality aspect. I just want to finish off by summarizing what we've talked about today, talking about this in vivo molecular imaging, talking about the animal models that we can use and the non-invasive nature of photoacoustic and ultrasound imaging, where we can do these longitudinal studies using targeting agents, and there are many under development for photoacoustics. I've talked about Optimios and nanohybrids who make their own nanoparticles, but there are many labs that are working on things like carbon nanotubes, liposomes, and other dyes, even fluorescent dyes and other things. The imaging itself, where we can get this very high resolution unmixed images at depth, and then we can quantify that signal to be able to show the accumulation and thus the molecular marker of interest, the expression of that molecular marker of interest. So I've described those advantages, the contrast agents, and I want to reiterate that the new software is going to be released this fall. If you're a current customer and you're interested in getting your hands on this software, please contact us. You can contact me directly or just go through our website, and we'll get you a copy of that in this fall. And now I'd just like to open it up to questions. Thank you very much for attending today.